For this particular chapter, we have talked about control and coordination in humans. We looked at the nervous system in the humans. We covered how action potentials happen. We also kind of covered the endocrine system in chapter 14 when we were talking about insulin, glucagon, and ADH. And to kind of summarize what we've covered, we talked about how the human body produces responses by using either the endocrine system or also the nervous system. You can kind of see the neurons are represented in the nervous system, and for the endocrine system, that diagram just shows you like hormones traveling inside the blood vessels. Just a very simple diagram over that. So what we have to cover right now is about plants, because here's the thing, plants do not have an endocrine system. They don't have a nervous system like humans do. So does that mean that plants are cursed to not produce any responses whatsoever? The answer is no, because plants themselves are able to produce responses depending on the situation. Like, for example, if you touch a particular plant called the touch me not plant, it will close. Plants will also grow towards light, that is called phototropism. Roots will usually grow downwards and shoots of the plant will grow upwards, that's called gravitropism, where it's responding to gravity. So plants themselves, interestingly enough, even though they don't have an endocrine system or nervous system, they do have things like electrical communication or also hormonal or chemical communication. And we are going to be looking at that in detail. So here's the thing, and I don't need you to memorize this part, by the way. So remember, action potentials are just the electrical impulse that are traveling along the neuron. And if you can see the neuron here, I'm just drawing part of the axon of the neuron or the nerve cell. And on the right side, I'm drawing a plant cell, which, they don't have an axon, right? Because plant cells don't have a nervous system. And you can see that the two plant cells are connected by a plasmodesma, which is the cytoplasmic connection between the two plant cells like that, which I've circled in green for you as a reminder. Now, the interesting thing here is as follows. Even though plant cells do not have the nervous system, just like the neurons in the humans, they also maintain a resting membrane potential where the inside is more negative and the outside is more positive. If you see the part where I've circled there in red, I made a slight mistake. I should have inverted the symbols. It should be negative in the cytoplasm and positive in the cell wall area. Made a slight mistake over there, so just pay that no mind because you don't need to memorize this. Now remember, in the humans, when they undergo the depolarization where the inside becomes more positive or a higher voltage than the outside, the charges will flip, and the flipping of the charge or the changing of the charge will cause it to depolarize the next area, the next area, and the next area. And this is called the wave of depolarization or the wave of action potentials. And this is just the electrical impulse moving along the neuron. The same thing also happens to the plant cells, because in the plant cells, if one area depolarizes, where the inside becomes more positive than the outside, it also causes the next area to depolarize as well, and it travels along the cell surface membrane of the plant cells. The only difference here is the depolarization is faster in the human because the axon is quite, straight. It's just a straight line. But for the plants, it is slower because it has to travel over a greater distance, and the cytoplasm will provide a bit of resistance. The plasmodesma makes it difficult for action potentials to travel, but the principle is very much still the same. There is action potential traveling along the plant cells as well. The only difference is it doesn't use a nervous system. It doesn't use neurons. But the principle remains the same. And you don't have to memorize the value during depolarization and repolarization for plant cells because it can differ depending on the species of the plant. So don't worry about that. So for this video, what we are going to be looking at is the electrical communication in plants, and more particularly, we are going to be talking about the Venus flytrap. Now, if you don't know what the Venus flytrap is, well, please Google it, just go and do a quick search on how the plant looks like. More often than not, students may not know the name of the plant, but they have seen this particular plant online and such. The interesting thing about this plant here is it has a kind of like a mouth over there, and we'll talk about that in detail later. And this Venus flytrap can actually ingest and digest insects and also small animals like frogs and such. Now, some students will immediately ask me the question, it's a plant, it should be able to do photosynthesis. So, oh, that's the thunder, by the way. Did you hear that in the background? So yeah, it's raining quite heavily over here. So the question is, if the Venus flytrap is a type of plant and it can do photosynthesis, why then does it have to ingest and digest insects? The reason is because Venus flytraps are carnivorous plants that grow in bogs and swamps and marshes, and these particular areas do not have enough nitrogen inside the soil. So what the plants will need to do is they need to digest the insects and the small animals to obtain the nitrogen from these animals. That's the reason why they do so. Okay, so the reason is they digest the insects to get the nitrogen compounds, and they use the nitrogen compounds to make amino acids and nucleotides that they need to grow and sustain themselves. And you need to know this, by the way. You need to be aware that Venus flytraps are plants that grow in areas with not enough nitrogen. Therefore, the insects provide the nitrogen the plants need. Usually, if it comes out in the exam, hopefully it's just a 1-mark question on the reason why Venus flytraps digest animals. Now, Venus flytraps have 3 parts that you have to understand. I'm going to break it down. Where the insect lands on that specific area of the Venus flytrap, the Venus flytrap will close. But if you notice, when they close, it doesn't close fully yet. And the third stage is when it closes fully. Okay, the trap closes, not completely in the second stage. And in the third stage, it closes completely. And this is where we have to talk about. This is what we have to talk about. We kind of have to explain how is it that when the insect lands on the Venus flytrap, it causes the trap to close. We have to cover that. And we also have to explain why it doesn't close completely. And we also have to explain what causes the Venus flytrap to then close completely and digest the insect. So, before we go through all of them in detail, we have to talk about the structure of the Venus flytrap. Now, a very important thing I love to ask my student is as follows. If I circle that particular area over there, what do you call that part of the plant? Some students will say, oh, it's the mouth. Other students will be like, oh, it's pink in color, so maybe it's the flower, perhaps. Another student just says, oh, it's a death trap. So, I mean, technically that's kind of, the third one is correct, by the way. But here's the interesting thing. That part of the Venus flytrap is actually a leaf. Okay, yes, it is a specialized leaf that kills animals, that is true, but that part is also classified as the leaf of the plant. So that's the first thing we have to understand. Now, I'm going to draw out the leaf over here, as you can see, and the leaf over here, you can see this pinkish color part, okay, that's the leaf, and that is called the leaf lobe. Okay, there are 2 leaf lobes on the top and at the bottom. The leaf lobes are convex. You need to know that word convex. Convex just means it bulges upwards. You can kind of see that at each part of the leaf lobe, it's kind of bulging upwards. Okay, that's what it's meant by convex. And to just give you a bit of an understanding as to what the lobe is, each of that section is referred to as a lobe. Okay, so the trap or the specialized leaf is made out of 2 lobes, and the 2 lobes are both convex. Now, another thing that we also have to know is each lobe at that pinkish area, it has 3 sensory hairs. And what's the function of the sensory hairs? The sensory hairs are to detect the animals or the insects that land on the lobe. And I will explain that further in detail. Now, on the edge of the lobes, they have stiff hairs. You have to be careful. Stiff hairs, the ones which are black in color, they look like needles. Those are not sensory hairs. They are just called stiff hairs. The sensory hairs are those ones which I've drawn out in red, and they are only located within the lobes as well. Okay, so you need to know the difference between the sensory hairs in the lobe and also the stiff hairs outside the lobe. As you can see in this particular diagram of the Venus flytrap, I'm just showing it to you. You can kind of see that this is the leaf lobe. I hope you can appreciate that it's kind of curved upwards. And when it's bulging or curving upwards, that means it is convex, and you can see the 3 sensory hairs over there within the lobes. And those 3 sensory hairs are to detect the animals and the insects that land on them. Now, another very important thing is I'm going to just circle a particular area over there, including the sensory hair and the leaf lobe area, and I'm going to draw out a few cells. As you can see here, all those cells are maintaining a resting membrane potential. The cell on the left, for example, is just connected to a sensory hair. That's not exactly how it looks like, but I just want you to just a simplified way of representing that the sensory hair is connected to the plant cells of the Venus flytrap, and each cell has a calcium ion channel, and within some cells of the Venus flytrap, they contain vesicles with digestive enzymes. Now, why do they need to contain vesicles with digestive enzymes? Well, obviously, once they ingest the insects, once they trap the insects, they have to digest it to get the nitrogen. That's kind of badass, isn't it? When it comes to the structure of the Venus flytrap, each leaf lobes are convex. Each lobe has 3 sensory hairs. The outer hairs are stiff. I've highlighted that as well. And within the plant cells of the Venus flytrap, they have calcium ion channels, and some of the cells have vesicles with digestive enzymes. So, how do they all work together? This is where we are going to put all of them in a hopefully seamless explanation. So, remember, the first thing that needs to happen is the insect must land on the Venus flytrap, and the trap must instantly close, although it must not close completely. Okay, so you can see that at the middle there's a bit of gap. Okay, it allows for a bit of gap. Now, some students will immediately ask me the question, shouldn't it actually close completely? Because it might allow the insects to escape. So there is a reason for that. So don't worry, I'm going to explain all that in due time. So the first thing that we want to cover is the stage of stimulation. Now, I'm drawing out the leaf lobe over here, and you can see each leaf lobe has 3 sensory hairs. Now, the first question I'm going to ask is this. The insect was just buzzing around, and it innocently lands at that particular spot, which I've put as a dot. Will the Venus flytrap close in this case? The answer is no, it will not close. Now, why doesn't it close at that particular area? Some students will immediately catch on and say, well, yes, it landed within the trap, but it did not stimulate the sensory hairs, or it did not touch the sensory hairs. That is why the trap does not close. Now, then I'm going to ask you the second question. The insect was just moving around, and it touches one of the hair. It just touches it once. Does the trap close now? Some students will say, yeah, in this case it will close. Here's the interesting thing. If the insect just touches the sensory hair once, the trap will still not get activated. There are a few conditions that need to be met for the trap to actually close. For the trap to actually get activated, the insect must stimulate the sensory hairs twice within 20 to 35 seconds. There is a range depending on the type of Venus flytraps, because there are different varieties as well. But for the purpose of the exam, you need to say, for the trap to close, the insects stimulate the sensory hairs twice within 20 to 35 seconds. You need to remember the time range. 20 to 35 seconds is good enough. Okay? Some students will ask me that, does it need to stimulate 2 different hairs, or can it stimulate the same hair twice? Both are fine. Okay? The insect can either stimulate 2 different hairs within 20 to 35 seconds, or it can stimulate the same sensory hair twice within 20 to 35 seconds. When the sensory hairs get stimulated, as I've drawn over here, the calcium ion channel opens and calcium ion rushes in. There is a calcium ion concentration gradient which forces the calcium ion to rush in. You don't need to go into that in detail. This has nothing to do with sodium ions per se. So when the calcium ions enter, it makes the inside more positive, which creates an action potential. So this is the stimulation part and the generation of the action potential. So what causes the closure of the trap? The action potential travels along the cells. Remember, I told you, in this diagram how the action potentials travel along the cell surface membrane through the plasmodesma. It starts traveling along the cells of the venous flytrap, and all you just need to mention here is the convex leaf shape, which is curved upwards, will become concave, which means it curves or bulges downwards, and that's what causes the trap to close. And this process happens rapidly. Now, we don't have to go into the detail of what causes the leaf lobe to change from convex to concave, because there are a few theories and explanations as to how this happens. There is a change in the elastic tension of the cell wall. Some modalities explain that there might be a rapid surge of water into the cell by osmosis. So there are a few things that we are still studying when it comes to the closure of the venous flytrap, so we are not going to go into the detail of that. All you just need to mention is the action potentials travel along the cells, which causes the leaf lobe to change from convex to concave rapidly, and this is what causes the trap to close. So this is the first two stages, where the insect stimulates the specialized leaf or the trap, and it causes the trap to close, not completely at first. Now, this is where we have to see stage 2 to stage 3, where the trap closes not completely, 2, the trap closes completely and digests the insects. Further closure and digestion. So what I'm drawing is, I'm just drawing the trap inside the trap right now, and you can see the insect within the trap. Now, if you were to be reborn as an insect in your next life, and you find yourself trapped within the venous flytrap, I hope you remember not to panic and try not to stimulate the sensory hairs any further. Because if you panic within the venous flytrap and you continue moving and continue, trying to get out of the venous flytrap and stimulating those sensory hairs, what will happen is it will cause the action potentials to further travel along the leaf lobe, and it causes the trap to completely close. And when it completely closes, in this case, there is no chance of escape at all whatsoever. So further closure is due to the insect further stimulating the sensory hairs, which causes the trap to fully close. There are no more gaps for the insects to escape from it. So remember, in your next life, if you're trapped in the venous flytrap, don't panic, okay? Don't know if you'll remember this advice, but let's hope, okay? Or don't go near a venous flytrap if you're an insect. What happens here is, as the insect further triggers the sensory hairs, as you can see, this is the insect over here. It is just flying and just buzzing around, and it might trigger the sensory hairs. It will cause the calcium ion channels to open. I'm not going to draw the depolarization. I'm kind of lazy to do the whole thing. But remember, as calcium ion goes into the plant cell, the cells will depolarize. It creates an action potential. But when the calcium ion goes into that particular cells with the digestive enzymes, just like what happens in the synapse, the calcium ion influx into the cell will cause the vesicles to move to the cell surface membrane, and the vesicles will release the digestive enzymes by exocytosis. So further stimulating the sensory hairs will cause the calcium ion influx, which causes exocytosis of digestive enzymes, and I don't need to tell you what happens to the insect. Gone, basically, right? So that's the end of the insect. Game over. So in that case, then the insect, the trap will usually remain closed for a few days. So what you just need to mention during the exam, if they do ask you a question on the closure of the venous flytrap, is as follows. The insect stimulates the sensory hair twice within 20 to 35 seconds. The calcium ion channels open and calcium ion rushes in, generating an action potential which travels along the cells of the venous flytrap. The leaf lobe changes from convex to concave rapidly, and then the insect further stimulates the sensory hair, which further closes the trap. The influx of the calcium ions going into the cell will also cause exocytosis of digestive enzymes to basically break down the insects and get the nitrogen. So that's what happens right there. Now, the thing that we have to cover is the adaptation of the Venus flytrap, because here's the thing. The Venus flytrap it's opportunistic because it has to capture the insects as fast as possible. But the weird thing is, if it's going to capture the insect, why does it need to stimulate 2 hairs between 20 to 35 seconds? Why not just stimulate 1 hair and immediately close? Isn't that better? You see, the stimulation of a single hair not causing an immediate closure is actually a type of adaptation. The reason is because sometimes what may happen is a drop of water from the rain might actually stimulate the hair. So the Venus flytrap wants to make sure that the stimulus is something that is tangible, that perhaps might mimic an insect. Okay, so if a drop of water or a debris or a leaf, a dead leaf just drops on the Venus flytrap and it stimulates 1 hair, the Venus flytrap will not immediately close because it doesn't have eyes. So in this case here, this is the adaptation to just make sure that accidental stimulation of 1 hair will not cause it to waste energy to cause it to close. Now, some students will then say, but teacher, the rain is not just a single drop. There might be a lot of drops of rain. So what if it does rain heavily? Surely more than 1 of the hairs will be stimulated between 20 to 35 seconds. Well, in that case, it may close, but it does not close completely. And the hairs are no longer stimulated because it's covered right now within the trap. Okay. Also, the gap, the reason why it doesn't close completely is because it wants to allow tiny little insects to escape. The reason why it wants to allow tiny little insects to escape is because it does not deem that these insects are worth digesting. Because think about it, keeping the trap closed and releasing digestive enzymes by exocytosis requires a fair bit of ATP. And if the meal is an extremely small meal, the amount of ATP spent digesting the small insect is not worth it. In fact, it might actually cause the Venus flytrap to waste energy in the process. So by ensuring that the trap does not close immediately, small for the lack of a better word, useless insects for the Venus flytraps will be allowed to escape. So in this case, insects having a small body or such and a small size would be at a selective advantage to survive. So there you go. So these 2 adaptations will allow the Venus flytrap to conserve ATP and prevent using ATP molecules unnecessarily in closing, trapping, and digesting the small meals. So we have covered the electrical communication aspect of the plant pertaining to the Venus flytrap. And in the last video for this chapter, we are going to be looking at the chemical or hormonal communication in plants.